Hello, welcome to the video. I Sai Shweta Pasma of Dhirubhai Ambani Institute of Information Communication Technology. As a part of our course curriculum, Element of Synthetic Biology course, under the guidance of Professor Manish K. Gupta, I have worked on synthetic biology and ICT. Um, how synthetic biology and ICT have helped each other in order to evolve as an emerging branch of science in the current generation. In this video, we'll be majorly focusing on DNA-based hard drives, their working and the reason for the development and the drawbacks. We'll cover all the things about DNA-based hard drives. As we have seen in our previous videos, what is synthetic biology, what are the major principles of synthetic biology, what is ICT, and why ICT for synthetic biology. We'll skip to the DNA-based hard drives. Uh, DNA-based hard drives. The major reason for the development of DNA-based hard drives is the exponential growth rate of data. As we can see that uh, as per the statistics given by the uh, uh, government, we can say that the data produced in the last 10 years is being currently produced in the 15 or 20 minutes, as everything is being digitalized and everybody is getting digitalized. And the current storage medium, like the magnetic and optical storage medium used, uh, are not uh, are not up to the mark or feasible with the current amount of data being generated, as because the magnetic medium storage only stores up to 10 GB per millimeter cube, 10 GB data per millimeter cube, and the optical medium storage stores only up to 100 GB per millimeter cube, whereas the DNA stores up to 100 EB per millimeter cube. Um, Next, the idea of DNA hard drives has been originated in 1999. The current requirement for archival storage led to the idea of DNA hard drives. That is, if you have followed news, you have known that the Facebook has just taken up an office to just store the hard drives for the used for archiving the data. Uh, as the hard disk or the magnetic optical medium stored occupies large space, whereas the DNA occupies very little space. For example, the like 1 pound of DNA can store almost 700 terabytes of data, which is more which is very revolutionary. The binary representation of DNA, as like the data is being represented in zeros and ones. In DNA, they are represented in nucleotides bases, that is adenine, guanine, cytosine, and thymine, that is AGTs, to generate a relationship between the binary and the nucleotide bases in DNA. The common platform is developed, and the adenine is represented as 0. The adenine represents a binary 0, cytosine represents binary 1, guanine represents binary 2, thymine represents binary 3, as per base 4 representation. For encoding uh, the data is encoded into binary digit in base 4, producing a string of n by 2 quaternary digits from a string of n binary digits. The quaternary digits are mapped to DNA nucleotides as follows. That is O to E, as I have told. For example, when you take a string 0, double one, 0, triple one, triple zero, 1 maps to 1301 base 4 string. DNA synthesis and sequence are prone to errors, requires a more careful encoding, which can be done by using encoding binary data in base 3 instead of base 4. Each ternary digit maps to DNA nucleotide based on rotating code shown in the given figure. This figure shows the binary 3 representation. In this encoding, this binary 3 encoding, the binary base 3 representation avoids homopolymers repetition of the same nucleotide. that significantly increases the chance of sequencing errors. Next, we have the diagrammatic overview of what happens in DNA. Overview: the DNA storage system consists of a DNA synthesizer that encodes the data to be stored in DNA, a storage container with compartments that store poles of data that map to a particular volume, and a DNA sequencer that reads DNA sequences and converts them into digital data. The basic unit of DNA storage is DNA strand, that is roughly 100 to 200 nucleotides long and is capable of storing 50 to 100 bits total. The main important point we have missed is we use we don't use the living cells in our body to store data, as the cells die after some time and the cells replicate, which leads to which can cause mutations, and the data can be changed due to the mutations or replications. So to overcome the problems, a research group in Harvard Medical School in Boston created DNA information archiving system that uses no cells. Instead, uses an inkjet printer that embeds short fragments of chemically synthesized DNA onto the surface of a tiny glass chip. The read and write process in DNA. You can see in the diagram how the read and write process in DNA is done. Uh, the storage medium needs a way to assign identification tags to data objects. So for a future retrieval of data, a simple key value architecture is chosen, where a put key comma value is a operation is assigned. Is assigned to the key. A mechanism to selectively retrieve only desired portion of a pool of data requires, that is random access required. We require a pair of PCR primes. As data is entered into the data, that is at the write time, those primes are added to the strands of data. At the read time, those primes are used in PCR polymerase chain reaction to amplify only the strands with desired keys, separating the DNA strands into collection of pools, balancing the trade-off between storage density, reliability, and performance. We use a library of reasonably sized pools. Cause a single pool would require many different primes to distinguish all the keys, which decreases the performance. Using a separate pool per key sacrifices the density excessively. Several algorithms are developed or used by the researchers for the efficient storage and retrieval of data in DNA. The main drawbacks in the DNA-based hard drive is high error rates. High error rates uh, because the PCR system is not being developed yet, and several algorithms have been used to get several algorithms are being developed to get to reduce the error rates, and the process is currently in development, and that is the researchers are working on that. And next, high cost of DNA sequences, that is unreliable for daily usage. Uh, Accessing or retrieving data in DNA is slow when compared to RAM. Uh, the coding units are very small, that is almost less than the size of a half a nanometer, where current transistors are hard to fit. So it requires more of the transistors need to be developed so that they can efficiently fit. So more of development and research is still required to put this actual DNA based hard drive system into actual usage, that is daily usage. Uh, a sophisticated protein engineering is required to reduce DNA replication. As the DNA replication leads to change of data, there should be a mechanism so that the replicated so that it when the replication is done in DNA, the data doesn't change, or if it changes, the changes should be noted. uh, Such an algorithm should be developed that if any changes due to replication occurs, it should be noted, or the replication should not affect the data, and design for security has to be developed as the current society is more towards the security of their own data. So the security system has to be developed. In actual DNA, anybody can access DNA. There is no lock or something. So uh, the researchers are actually working on the security issues present. It is currently not a feasible method, but further research uh, will make it possible for the use of everybody uh, to get into practical usage. So we need to wait for we need to wait for 10, almost nearly a decade, for this system to come into actual practice, which will make the world revolutionary. Finally, thank you so much for watching the video. For any queries, you can contact 2014. You can mail us. You can mail me at 2014014040 @diict.ac.in. Thank you.